Hi guys, chapter 8. So we are starting to apply what we learned from chapter 7 onto chapter 8. So there are concepts coming back in addition to new concepts um, coming your way. So chapter 8 is also very, very, very interesting. Um, Chapter 7, we saw electrical signals being generated and being transmitted within neurons, within each neuron. So on the dendrite and the cell body, the neuron can generate graded potentials, for example, EPSPs and IPSPs. The axon hillock generates an action potential, and the axon propagates the action potential down the axon. When it hits the axon terminal, it induces the release of chemical messengers by the axon terminal called neurotransmitters. So that's chapter 7. Chapter 8 will pick up from there. So once the chemical messengers, these neurotransmitters, are released into the synapse, into the synaptic cleft, by simple diffusion, they migrate through and they bind to receptors on the next neuron. So once that happens, when the chemical messengers diffuse across its synaptic cleft and binds to receptors on the postsynaptic neuron, that process has a new name. We call that synaptic transmission. So when we say synaptic transmission, we are talking about communication between at least two neurons, right? So it takes two to tango here. So in order to have a synapse, you need to have at least two neurons, two cells, two nerve cells, right? So when we talk about synaptic transmission for the rest of the semester, we imply that we're talking about interaction, communication between at least two neurons, presynaptic neuron, postsynaptic neuron. And once we talk about synaptic neuron, meaning interactions between neuron, communications between neuron, it gives rise to something amazing. And we call that integration, neural integration. And you think about it, when you think about the nervous system, even though it is the most complex organ system because it controls everything else, but it gives rise to thought, right? So abstract concepts. But amazingly, when you boil it down to its core, the nervous system has only three functions. It has a sensory function, it has a motor function, and it has integration as a function. So sensory means it has the ability to receive information. Motor, its ability, it's a reflection of its ability to send out a decision. But before it can send out a decision, it needs to digest the incoming information. So sensory information comes in. That sensory information needs to be digested, needs to be thought over. That process, we call it integration. Once that's done, it shoots out its decision. We call that a motor command. That's the entire organ system. And more amazing is the nervous system is the only organ system where a single cell is capable of performing all the function of that organ system. In other words, a neuron, a single neuron can do all of these three fundamental functions of the nervous system. So a neuron can receive information. Sensory information arrives at the dendrites or the cell body. Integration occurs at the axon hillock. And the motor decision, that's the action potential propagating down the axon, and then the release of the neurotransmitter. So every time a neuron releases its neurotransmitter, that's a motor decision. That's the beauty and the power about the nervous system, is a single cell is capable of performing all three functions of the entire organ system. Again, only the nervous system is capable of doing this. All the other organ system, you don't see that. Only the nervous system, you see it. And this is priceless information. No one tells you this. No textbook will tell you this, right? So this is based on years of experience. And this is integration on my own because I'm writing a book. So that's how you should understand the nervous system. From a single cell all the way to the entire organ system, you have only three functions: sensory, integration, motor. Okay. So for the learning objective, we're going to go over again, same three pillars. We're going to revisit the anatomy, the functional anatomy of a synapse. So we're going to go over electrical synapse and chemical synapse again. We're going to also visit what is in front of the synapse, what is after the synapse. So before the synapse is the presynaptic neuron. After the synapse is the postsynaptic neuron. So when we say synaptic transmission, we are talking about that. So synaptic transmission involves presynaptic and postsynaptic neuron, at least two neurons. And then once the chemical messenger binds to the receptor, what happens? So we're going to go over structure function correlates. I'm going to go over the signal transduction mechanism. What happens once the chemical messenger binds to its receptor? So the receptor is physically located on the dendrite of the postsynaptic neuron, or the receptor is located specific on the cell body of the postsynaptic neuron. When I say cell body, I'm excluding the axon hillock, okay? And when I say on the dendrite, I'm talking about the receptor. It's a transmembrane protein located at the plasma membrane, located at the cell membrane, located at the phospholipid bilayer of the dendrite of the postsynaptic neuron, of the cell body of the postsynaptic neuron, again, excluding the axon hillock. Once the neurotransmitter binds to that receptor, that transmembrane protein acting as a receptor, what happens? That will bring in the signal transduction mechanism, the signal transduction cascades, the events that basically follows after the interaction between the neurotransmitter binding to its specific receptor. And when we talk about neurotransmitter, we learn about this. There are neurotransmitter that will induce the postsynaptic neuron to generate excitation signals, we call EPSPs, or induce a postsynaptic neuron to generate inhibition signals, we call inhibitory synapses. So we're going to revisit them as well. So what are the excitatory neurotransmitters and what are the inhibitory neurotransmitters? And what are the consequences of this? And we know that already, right? So excitatory neurotransmitter are acetylcholine, ACH, ATP, glutamate, and serotonin. Inhibitory are GABA and glycine. We learned that, but now in chapter 8, we learn the details about it. What happens when they bind to the receptor? right? Why is it some of the neurotransmitter will cause induce EPSPs and others cause IPSPs? Why is it that ATP, glutamate, serotonin, and acetylcholine induces depolarization, whereas GABA and glycine induces hyperpolarization? That's the gist for chapter 8. So the details we get to appreciate. And lastly, under homeostasis, we tied everything together. We go in and we focus on integration. And specifically, we're going to go over a case where we say we learn about presynaptic modulation. So all of this should be a beautiful, gorgeous 90 minutes of lecture about chapter 8. So with that said, let's get this thing started, starting with the first slide. So synaptic transmission, again, requires at least 2 neurons. And when we say electrical synapse, we are talking about the 2 neurons are physically contacting each other. So they're physically touching one another through gap junctions. So gap junctions are 24/7 openings between 2 neighboring cells. In this case, cell 1 is a neuron, and cell 2 is also a neuron, right? So electrical synapse, because synapse is a microscopic space between 2 neurons. N stands for neurons, right? This looks like a Z, it's a number 2. Sorry. Okay. So direct communication via gap junctions. Some neurons, we have this, electrical synapses. The problem with this is there's no directionality. So the electrical signals can move back and forth. So this the downside is, the caveat is there's no directionality. The advantage is it's extremely fast. So through these electrical synapses, communication goes much, much faster than synaptic communication, synaptic transmission. Well, because synaptic transmission, which on the next slide, is because there's a space between the communicating neurons. So when there's a space, it takes time for the neurotransmitter to diffuse across. So every time you have a synapse, you basically slow down communication between neurons. But if you have an electrical synapse, there's no such diffusion. So the message is almost instantaneous. But there's no directionality. So there's a balance that you need to take into account. Chemical synapse, this is basically the majority of cell-cell communication is through synapses. So advantage of chemical synapse over electrical synapse, directionality. Disadvantage, chemical synapse compared to electrical synapse, chemical synapse are slower. Now, when we say chemical synapses, there's a synapse, there's a microscopic space between the 2 neurons, and there are chemical messengers called neurotransmitters, right? So the neurotransmitters are released from a presynaptic neuron. So this is a presynaptic neuron, the neuron before the synapse. And you have a postsynaptic neuron, the neuron after the synapse. And this is a synapse. You say synapse, or you can say synaptic cleft, means the same thing in this context, right? So synapse or synaptic clefts means the same thing. the synaptic vesicles contain the neurotransmitter. So the neurotransmitter are the chemical messenger. So each triangle is one neurotransmitter molecules. For simplicity, the synaptic vesicle here is showing containing 4 neurotransmitters, but in reality, it's much more than 4 within each synaptic vesicles. And the action potential arrives here. So this is the AP. It arrives, it depolarizes the membrane, and that depolarization is picked up by a voltage sensitive calcium channel. Now again, change would gate it to sensitive, right? So this is voltage sensitive calcium channel. So the action potential, the depolarization carried by the action potential induces the opening of that voltage sensitive calcium channel. And calcium, massive influx, shown by the black arrow here. When calcium, the massive calcium influx, it induces the vesicle to fuse with the cell membrane, to fuse with the plasma membrane of the axon terminal. So this structure here, we're going to say that AT stands for axon terminal, right? So the vesicle, the synaptic vesicle fuses with the membrane, the cell membrane, the plasma membrane, the phospholipid bilayer of the axon terminal, and allowing the vesicle to dump its content, which is neurotransmitter, into the synapse. And the neurotransmitter passively being transported across the synaptic cleft. And the mechanism of transport is called simple diffusion. And once it goes on here, it will bind to the receptor on the plasma membrane, on the cell membrane, on the phospholipid bilayer of the postsynaptic neuron. So this area here can be a dendrite. Or the cell body, soma. Right? Dendrite, D-E-N-D-R-I-T-E. D-E-N-D-R-I-T-E. Dendrite or soma, S-O-M-A. Soma is the same as cell body. And when I say soma or cell body, I'm excluding the axon hillock, right? And this dendrite or soma will generate either EPSPs or IPSPs, depending on the type of the neurotransmitter that binds to the receptor. So when I say receptor located on the postsynaptic neuron, I'm talking about these are transmembrane proteins, TMP. So transmembrane protein, meaning the protein that are located on the cell membrane, on the plasma membrane, on the phospholipid bilayer of the postsynaptic neuron. Specifically, dendrite or soma of that postsynaptic neuron. Okay, so here What you have here is this blue space here is your synapse or synaptic cleft. This here is the cell membrane or the plasma membrane or the phospholipid bilayer. So PM stands for plasma membrane. Plasma membrane, for example, of a dendrite. Okay, or plasma membrane of the soma on the postsynaptic neuron. I repeat, plasma membrane of a dendrite of the postsynaptic neuron, or plasma membrane of the cell body belonging to the postsynaptic neuron, excluding the axon hillock. And here you have a transmembrane protein, TMP, used as a receptor, right? And not only that, the receptor has a tunnel, a pore, a passageway called a channel. So this receptor is also an ion channel. When it opens, it will flux ionic species. Now we learned that from previously, right? Okay, so over here, you see the neurotransmitter. So this is one neurotransmitter molecule. So the neurotransmitter binds to the receptor. So this is the binding site for the neurotransmitter on the receptor. So this is like the key and the receptor is a lock. So this interaction is exquisitely specific, right? Acetylcholine binds only to the acetylcholine receptor. Glutamate binds only to the glutamate receptor. Serotonin binds only to the serotonin receptor. ATP binds only to the ATP receptor. GABA binds only to the GABA receptor. Glycine binds only to the glycine receptor. So this type of receptor, we can use the word gated. So they are called ligand or ligand gated, or neurotransmitter gated, or chemical gated channels, specifically ion channel. And when we say the receptor, if it has a channel, we call it channel linked receptor. It will be called ionotropic receptor. Ionotropic means fast response. The word ionotropic means fast. So the response occurs on a millisecond time scale, faster than a blink of an eye, right? So this is extremely, exquisitely fast, quick. We're gonna talk about a slow response later. It has another name. I don't want to jump the guns here, but for now, ionotropic receptors means ion channels. Same thing. You can say ionotropic receptors. We are talking about ion channels. And when we say ion channels, we can have two types of ion channel. We can have a cation channel, and we can have an anion channel, right? So the cation channel fluxes positively charged ionic species. Anion channels flux negatively charged ionic species. And what is also unique about the gated neurotransmitter gated ion channels is when they open, they flux multiple ionic species. So when we say ligand gated, and especially the ligand gated or neurotransmitter gated cation channels, meaning they flux positively charged ionic species. So you should have 3 arrows here and not 2. There's 1 missing. I'm going to do it all also in green. So there are also another one coming in. So there are 2 influx and 1 efflux. Yeah. So I'm going to write in black so we can clearly see. This here, that's for sodium. So we have a sodium influx. This one here, that's for calcium influx. Calcium carries 2 positive charge. So this is a divalent cation. Sodium is a monovalent cation. And then this green that comes out, that's potassium efflux. Potassium is leaving the postsynaptic neuron. Sodium goes in the postsynaptic neuron. Calcium goes in the postsynaptic neuron. But potassium is leaving the postsynaptic neuron. So potassium is effluxing. So efflux, E, F, F, L, U, X, right? Efflux, E, F, F, L, U, X. Both calcium and sodium, they influx. They go in. INFLUX. You see this only for the cation channels, the ligand-gated cation channels, because the voltage-sensitive ion channels, they flux only 1 ionic species at a time. Voltage-sensitive sodium channel, influx only sodium. Voltage potassium channel, efflux only potassium. Voltage sensitive calcium channel, influx only calcium, right? So the 3 families of voltage sensitive ion channels: voltage sensitive sodium channel, influx is only sodium; voltage sensitive potassium channel, efflux is only potassium; voltage sensitive calcium channel, influx is only calcium. But the ligand gated or neurotransmitter gated cation channels flux all 3 ionic species at the same time through the same channel, right? Those ligand gated cation channels, influx calcium, influx sodium, and efflux potassium through the same pore, through the same passageway. And that's unique to the ligand-gated, neurotransmitter-gated cation channels, because even the ligand-gated anion channels don't do that. So if you talk about the anion channel, meaning they flux negatively charged ionic species, anion, A-N-I-O-N. So what you're seeing is, all this goes out, that goes out. You have to imagine this. I cannot erase it. So imagine a disk, ignore this. Ignore the arrow that goes out. There's no arrow going out. There's only one green arrow going in, right? And that's for chloride, negatively charged chloride, influxing. And only chloride fluxes in through this channel. Now we also have leak channel, right? So or other type of channel that also fluxes only, for example, potassium. So there's a type of channel that you're going to learn later this lecture that only fluxes potassium, meaning specifically allowing only potassium to leave. And that concept you saw already, the leak channel, the leakage channel, involved in setting up the resting membrane potential. So the sodium leak channels only influxes sodium. The potassium leak channel only effluxes potassium cations. But these are not gated. These are always open. So that they're not on the same concept. These guys we're talking about, they can be opened by a chemical messenger. In this case, a neurotransmitter, right? So that's a fast response. So again, when we say fast response, it goes through ionotropic receptors, meaning ion channels, right? Ionotropic receptors, we talk about ion channels with a pore, with a passageway, right? This is a closed channel. This is an open channel, okay? Now we move on to the other extreme, slow response. And the slow response occurs on a slower time scale in terms of seconds instead of milliseconds. It can also be more sustained. It can, go over a longer time frame, but the ionotropic lasts only within a few milliseconds. And there's a typo here. This should have been an E. Metabotropic, so ME, okay? This is not an L, this is an E. I'm gonna change that capital so you can see clearly so there's no misunderstanding here. E. Metabotropic. In other words, the G protein receptors, they do not have a pore. They are not ion channels. So here is your metabotropic receptor. So this is the receptor. And the receptor is associated with another protein. And that protein is called a G protein. So that's why I say G protein linked receptor. Yeah. So when we say G protein linked receptor, we are talking about this receptor here. So G protein linked receptor is this one and this one. And the G protein has 3 subunits. You have the alpha subunit, you have the beta subunit, and you have the gamma subunit. For this class, we're going to focus on the alpha subunit. When I say subunit, it's spelled S-U-B-U-N-I-T, right? Subunit. So the G protein consists of 3 subunit. They're all proteins. There are three interacting protein. One is called alpha, another one is called beta, the third one is called gamma. Each one has unique functionality. So we're going to focus, especially for today's lecture, on the alpha subunit. So when the neurotransmitter shown here, so this is the neurotransmitter. Right, exquisitely specific, binds to the G protein coupled receptor. So the receptor is in blue. So that's the exquisite specific binding site, only compatible to the neurotransmitter. So the binding of the neurotransmitter to the receptor turns on the receptor. So this is your receptor, right? So upon the binding of the neurotransmitter, it activates the G protein. The G protein becomes activated, specifically focusing on the alpha subunit. And the alpha subunit, once it's activated, it can go on to open an ion channel. So this alpha subunit can open an ion channel or can induce the closure of an ion channel. So this ion channel has nothing to do with the G protein coupled receptor. So I keep saying couple. When I say linked here, G protein linked receptor, it is also called coupled. Right? G protein coupled receptors. This is not millisecond, this is more like seconds. Right? Seconds to hours. Now, this slow response is called direct coupling. Meaning the G protein directly lead to a response, meaning it opens an ion channel, right? When the channel opens or closes, it induces the cell a direct response. We call it direct coupling. But oftentimes, that alpha subunit will not interact with an ion channel. It will interact with another substrate. It interacts with another protein, like an enzyme, shown in this panel here. So in this case, it's the same phenomenon. Now, the alpha subunit here will activate a membrane-bound enzyme, right? So this is another transmembrane protein acting as an enzyme, right? So this enzyme here is a transmembrane protein. And we get to learn that later today. That enzyme specifically has a name, AC, adenylyl cyclase. Don't write it down, just listen. So the adenylyl cyclase will convert ATP to one of the most important second messenger, cyclic AMP. Just down here, second messenger. It's a new concept. Because the primary, the first messenger, is water soluble. So it will never get inside the cell. It remains on the outside. The neurotransmitter, in this case, this neurotransmitter, when it has a receptor of the cell membrane like this, it means one thing. That neurotransmitter was never allowed to cross the cell membrane. So that neurotransmitter must be water soluble. It's not compatible with the phospholipid bilayer. That's why it needs a translator, if you will. And that translator is at the surface of the cell in the form of a receptor. So that messenger, the first messenger, the neurotransmitter, remains on the outside. So its message is conveyed inside by the production of second messengers. And the second messengers can go on to carry out function. For example, this second messenger here, once produced, can induce, can affect the function of an ion channel, right? Again, can open or close an ion channel that is located in its proximity. Same here. The difference between this one in panel B, the alpha protein activates an ion channel, whereas panel C is the second messenger that activates the ion channel, right? So IC stands for ion channel. IC stands for ion channel, right? Both cases, B and C, involves the G protein coupled receptor. So the receptor and the G protein, both cases. Upon binding, in this case, panel B, alpha subunit directly activates an ion channel. Panel C, the alpha subunit indirectly activates an ion channel because the alpha subunit activates an enzyme. The enzyme produces a second messenger, and the second messenger activates an ion channel, right? So this is, you can say this is indirect coupling, and this is direct coupling. So that's the two signal transduction mechanism, the fast and the slow response. And I almost forgot, when we say slow response, the slow response is mediated by, like I said here, metabotropic receptors. And metabotropic receptors are not ion channels, okay? So they are G protein coupled receptor here. There's no ion channel to be seen anywhere, okay? So make sure that you have that down, right? So there's no pore, no opening within that receptor. And they are much slower to respond than an ion channel. Now, once the neurotransmitter binds to the receptor, it can induce the postsynaptic neuron. It can basically change the resting membrane potential of the postsynaptic neuron. It can either depolarize it or it can hyperpolarize it, right? So when it depolarizes it, we call it excitatory signals. So the signal is EPSPs. So when you say, when you look at this, excitatory, that's EPSPs, right? Excitatory postsynaptic potentials, EPSPs. The E here stands for excitatory. So excitatory postsynaptic potentials. This is done by channel opening. So when you open a channel, right? So these neurotransmitters, there's one missing. I'm going to add the fourth one. So acetylcholine, ACH, glutamate, ATP, and the fourth one, serotonin. Okay, serotonin, serotonin, serotonin. These four neurotransmitters, they have receptors that are all ion channels, right? So they all lead to fast responses, ligand-gated channels, right? So ligand-gated channel means ligand-gated ion channels, ligand-gated cation channels, shown down here. So the neurotransmitter, they can be of four types. They can be ACH, they can be glutamate, we can talk about ATP, or it can be serotonin. So these four examples are down here. They bind to each specific receptor, and when they bind, again, there should have been three arrows here, right? So there are two influxes, sodium and calcium they go in, potassium is effluxing. So there should have been a third arrow going in. And that third arrow is calcium. And what's going to happen is, and this is important here, inside the cell is always less positive. So, but to show that, we write negative sign here. Right, at rest, inside is more negative than outside. That's why the resting membrane potential is negative, because negative is inside. So at rest, resting membrane potential is minus 70 millivolts. When you open this channel, there's a net influx of positive charge. Even though potassium is leaving, there's less positive charge leaving than positive charge entering, right? So the efflux of potassium is more than offset by influx of both sodium and calcium ionic species. So at the end of the day, what you have is inside the cell, there's a net gain of a lot of positive charge versus smaller positive charge out here. What's going to happen is around this area now you have flipped the polarity around this area, and out here you have less positive charge indicated by negative sign, right? Now you just flip the polarity, and this is called a depolarization signal. You just depolarize the cell. You just flip the polarity of the cell. So depolarization. Depolarization, Depolarization. Depolarization means you're just flipping the polarity of that particular location on the cell membrane. Okay. And basically what it means is the cell membrane no longer at minus value, it will move toward a positive value, for example, plus 60 millivolts. Okay. So that's how you generate EPSPs. Now, 60 millivolts is very large. That's one action potential. Maybe to a positive value of, let's say, around 20 millivolts. We're talking about dendrites in cell body here. We're not talking about axon hillock yet. Okay, so that's leading to EPSPs, and these will go toward the axon hillock, and this signal dissipates with distance, right? So EPSP dissipates with distance. So we learned that from chapter 7. We applied here. So again, this cell membrane, it might be at one of the thousands of dendrites or the cell body, which is closer to the axon hillock. So if it's the cell body, then chances are it will get to the axon hillock and the signal is still there, right? Less in terms of intensity, but still there. If it's out, the farthest out of the dendrites, then most likely a single EPSP will die out before it even gets close to the cell body, right? So that's why you need a lot of EPSPs being generated almost simultaneously or within the same closer time frame, so they can be added on top of one another, helping to increase the depolarization enough juice, enough steam, allow it to spread all the way to the axon hillock. Otherwise, it will die out before it gets there. So by directly opening an ion channel, you can depolarize the membrane. So this is one way. So through direct channel opening, right? So fast response through ion channels. There's another way. Again, EPSP, but now it's through a slow response, through a G protein coupled receptor. And we saw that already too. This is also leading to an EPSP, but the mechanism, the transduction mechanism is slightly different. And we mentioned that as well. Let me erase this. Change the color. So you have the neurotransmitter binding, the G protein coupled receptors here. So upon binding, the G protein is activated. Its alpha subunit activate a transmembrane protein acting like an enzyme. This is the enzyme that I mentioned earlier. So this is the adenylate cyclase acting. This is a bona fide enzyme. And that enzyme, E stands for enzyme, converts ATP, the same cellular energy molecule, into a very important second messenger, cyclic AMP. This cyclic AMP is your second messenger, SM, right? One of the most important second messenger, cyclic AMP. Cyclic AMP goes on to activate a protein kinase. This is another enzyme. Protein kinase means its ability to phosphorylate a substrate. In this case, protein kinase A adds a phosphate group to a potassium channel. And what's going to happen is, upon phosphorylating, the potassium channel, this potassium channel shuts down. It closes, right? And this is important. Potassium channel close. And there are consequences. So when the potassium channel closes, potassium is no longer leaving. The inside of the cell accumulates a positive charge. And this is new here. The accumulation of positive charge upon the close of this potassium channel will also lead to the membrane depolarization. This is also depolarization signal. Okay, depolarization. So this is another mechanism of depolarizing the cell. by shutting down the potassium channel. Because when the potassium channel opens, potassium effluxes. Potassium is leaving the cell. When you close down, potassium is no longer being able to leave the cell. Potassium accumulates inside. So we have a massive accumulation of positive charge here, less positive here, because potassium is not leaving. We have a depolarization signal. So both cases leading to EPSP by different mechanism, right? This one is by depolarizing the cell through net positive charge influx, whereas this one generating depolarization but preventing potassium positive charge efflux. So in both cases, when you accumulate a net gain of positive charge on the inner leaflet of the membrane, it will depolarize the membrane. Again, one more time. Every time that you have a net accumulation of positive charge on the inner leaflet of the membrane, that's a depolarization signal. And that net gain of positive charge on the inner leaflet can be created in two different mechanisms. Mechanism number one, channel opening, allowing a net influx of positive charge, or another mechanism, channel closure, in this case specifically potassium channel closure, preventing potassium efflux, preventing potassium from leaving the cell. Therefore, potassium, which carries a positive charge, accumulate on the inner leaflet. It's also a depolarization signal. Okay, so those are two ways of generating EPSPs: by opening an ion channel or by closing of ion channels. Moving on to the next topic. Now, how does the neuron generate IPSPs? So we talk about EPSPs. Now we're going to talk about IPSPs. And IPSPs, we are talking about ion channels that flux only chloride, right? So chloride channels. So this is only fluxing one ionic species, very similar to the voltage sensitive channels. So the neurotransmitter, it can be GABA or glycine, right? So the neurotransmitter here, each circle is one neurotransmitter molecule, right? So it can be GABA or glycine. GABA is released by neurons in the brain. Whereas glycine is released by neuron in the spinal cord, SC spinal cord. So when it binds, it opens a channel, chloride comes in. And what happens is, when you have a net accumulation of negative charge, you're making the cell, the inner leaflet, even more negative than before, right? So here, this is hyperpolarization. You are making the polarity worse than before. You're making the inner leaflet even more negative than when the neuron is at rest. So for example, instead of being negative 70 millivolts, following IPSP, the membrane potential can be minus 90 millivolts, for example, or even more negative, minus 100 millivolts, right? That's called hyperpolarization, meaning that signal, if it gets to the axon hillock, it will prevent the axon hillock from firing an action potential. Okay, let me repeat. If that EPSP gets to the axon hillock, most likely the axon hillock will not fire any action potential, because that EPSP will bring the resting membrane potential farther away from the threshold potential. So the axon hillock will not be able to get to the threshold potential so it can fire an action potential. So that's one way of creating an IPSP. There's another way of creating an IPSP, and that's on the next slide. So this is, there's yet another mechanism. So there are two ways to create IPSPs, and there are also two ways to create, EPSPs, and now we're going to learn there's also two ways to create IPSPs. And before I get to that, when you have a chloride accumulation in here, the cell needs to clear it, and that requires energy. The cell spending energy, ATP, to pump out chloride outside. So this is to bring back homeostasis, right? So incoming chloride will be kicked out. When it kicks out, it's at the expense of energy. So this side is active transport, AT, active transport. It requires energy. But this side is passive transport, more specifically facilitated diffusion. So it's a passive transport mechanism, not requiring energy. This is driven by concentration gradient of the chloride being more on the outside drives in. But this one requires cellular energy. So this is active transport versus passive transport. Right? Passive transport. A specific example of passive transport, facilitated diffusion. So let's go on to talk about the second mechanism to generate IPSPs. Again, the neurotransmitter binds to an ion channel, and this channel that I mentioned earlier is unique in the sense it fluxes potassium, and more specifically, it effluxes only potassium. So the cell is losing positive charge, right? So here, positive charge is on the outside, little positive charge here. So what you're making here is inside the inner leaflet, you have even more negative than before, because the cell, the inner leaflet is losing a lot of positive charge. So this here, again, is hyperpolarization signal. You're generating IPSP through potassium efflux. Okay. So EPSP, you can generate IPSP by potassium efflux, or you can generate IPSP by chloride influx from the previous slide, right? So potassium efflux or chloride influx will generate IPSPs, will hyperpolarize the cell membrane, the plasma membrane, or the phospholipid bilayer of the dendrite or the cell body of the postsynaptic neuron. When I say cell body, again, I'm excluding the axon hillock. So those are the four mechanisms by which this neuron, the postsynaptic neuron, can generate EPSPs or IPSPs. So two ways to generate EPSPs, two ways to generate IPSPs. And I guarantee you, they will be assessed during both the lecture exam and the lab test. So no surprise here. Every semester, these concepts are so fundamental to this class, they will be asked during an exam or a lab test. So don't tell me, don't blame me that I did not warn you about this. So make sure that you understand this, because all these concepts will come back in Unit 3 when we talk about the cardiovascular system. So it is fundamentally important, and also skeletal muscle tissue contraction. So all of this will come back. Now, moving on to neural integration. So the mini brain of the neuron, the axon hillock, right, which is part of the cell body. It's a delta-shaped structure connecting the cell body to the axon, and it can integrate. It can add all incoming EPSPs. It can add all incoming IPSPs, and it will compare that with the threshold potential. And the threshold potential, when we say threshold, it means the threshold potential at minus 55, right? So the cell membrane, the plasma membrane, and the phospholipid bilayer at the axon hillock, do I have minus 55 or more positive? Do I have at least minus 55? If I have minus 55 millivolt, I can fire an action potential. If I have minus 54 millivolts, I can fire an action potential. If I have minus 56 millivolts, then I remain silent. At least minus 55 or more. So minus 55, minus 54, minus 53, minus 52, minus 51, minus 50, all of these will fire an action potential. But minus 56, minus 57, minus 58, minus 59, minus 70, nothing happens. Okay? Now, it can add on a time scale, so temporal summation, or it can add spatial summation, so coming from different parts of the postsynaptic neuron, right? So if, for example, if I'm pay attention to only for neuron A, over time, that's temporal. Or only B over time is temporal. Only C over time is temporal. But when I say spatial, then it comes from different locations. So spatial means if I'm adding the signal coming from A, EPSP is created by the signal coming from A, EPSP is created by signal coming from B, and IPSP is created by signal coming from neuron C. So 3 different locations, we call that spatial summation. So in reality, you can have both simultaneously, temporal summation and spatial summation, right? So temporal summation is same neuron over time, spatial summation, different neurons at the same time or over time as well. The point is that axon hillock, when it adds all of this up, does it get to threshold? If it gets to threshold, it fires action potential. If it doesn't, then nothing happens. So over here, let's begin with temporal summation. temporal summation here. So the same neuron A fires over time. Okay, over this here. So it fires once, it dies out. It fires another one, it dies out. So it cannot add it up. But here, it fires so close to one another that the two EPSPs can add it up, can help each other to get the threshold. Axon hillock fires an action potential, right? So this here, that's an AP action potential that was generated at the axon hillock AH. This one, AH. And the signal was created here on the cell body, right? And it gets to the axon hillock, allow the axon hillock to fire that action potential. If it's this area, because these two areas, this one and this one, no AP being generated because it never got to threshold. Okay, so that's temporal summation. You can do the same thing for B, you can do the same thing for C. Now going over spatial summation, so we are talking about different neurons here. So A fires by itself. Nothing happens. B fires by itself. Nothing happens. C fires by itself. It's even worse. Nothing happens because that's hyperpolarization here. You see that goes away from threshold. Okay, but if B and C fires the same together, you have it over here. The two adds up, you have an action potential here. Generated at the axon hillock. This is the axon hillock. Right. So if A and B fire at different location, but close enough in time too, they add up, because here they fire, A and B they fire, but they're so far apart they can't add up. So they're different location, but they're not at the same time. Here, the different location at the same time, then they can add up and you have boom, action potential. Okay, so we call that summation, the ability of an axon hillock to integrate different incoming signals over time and over different, coming from different presynaptic neurons. So temporal summation versus spatial summation. So again, one more time, spatial summation means the signal comes from different neurons. And when we say neuron, let's make sure that we understand this. A, B, and C are three pre-synaptic neurons. Right? Three presynaptic neurons compared to the yellow neuron. This is a postsynaptic neuron. So this here This yellow neuron is the postsynaptic neuron. Postsynaptic neuron. Okay, presynaptic, P-R-E-S-Y-N-A-P-T-I-C. Postsynaptic, P-O-S-T-S-Y-N-A-P-T-I-C. Frequency coding means how many signals are being generated per unit time. So we're going to come back to that frequency coding when we talk about sensory physiology. So the number of electrical signals that are being generated per unit time, for example, an action potential, 5 action potential per second versus 20 action potential per second codes for different types of information. We call that frequency coding. It's important because the action potential is always the same, right? It has the same intensity. So in order for neurons to understand the signal, the meaning, information coming with that signal, it has to be how many action potential arriving per unit time. That's how the brain understand information, because there's no way other way. All action potential are the same. So like every single one of us had the same shirt, right? Every single one human being is the same size, identical. So how do we know that? So in order for the brain to understand that, so it's gonna say, okay, it recognizes how many action potential it receives per unit time. So are we talking about 1 action potential per second, 5 action potential per second, 20 action potential per second, or 50 action potential per second? Each one contains a unique, distinctive piece of information. We call that frequency coding. Moving on to the last slide. Presynaptic modulation. The name tells you where the modulation occurs. It is affecting the presynaptic neuron. And then the presynaptic neuron, once being affected, it will basically also affect the postsynaptic neuron, right? So in this case here, the postsynaptic neuron is neuron X. Neuron X and neuron Y are postsynaptic neuron. Neuron DC or presynaptic neuron. Neuron GF or presynaptic neuron. Neuron E and neuron H, that's the neuron that modulate the presynaptic neuron, right? So neuron E here modulates the presynaptic neuron C. Neuron H modulates the presynaptic neuron F, right? In this case, the neuron E, the axon terminal of neuron E interacts with the axon terminal of the presynaptic neuron C. So in this case here, when we say presynaptic facilitation, neuron E induces neuron C to release more neurotransmitter. So neuron C increases the release of its neurotransmitter. Increased neurotransmitter means also you increase the intensity of the graded potential. So on the membrane, on the cell membrane of the postsynaptic neuron X, you can generate more EPSPs if the neurotransmitter is excitatory, right? So more ATP release, more EPSP produced. More glutamate release, more EPSP produced. More ACH release, more EPSP produced. More serotonin release, more EPSP produced. Down here, we talk about presynaptic inhibition. So now the neuron H in pink prevents neuron F to release its neurotransmitter. So here we have less neurotransmitter. Less neurotransmitter means less EPSPs, less ATP, less EPSPs, less glutamate, less EPSPs, less serotonin, less EPSPs, less ACH, less EPSPs, right? So this, we call that presynaptic and facilitation and presynaptic inhibition. And both cases involves a concept that you learn from anatomy. The interaction is called axo-axonic. So that's between here, between two part of the, so axo-axonic, from the axon E to axon C. Specific in this case, axon terminal of neuron E interacting with axon terminal of neuron C. Axon terminal of neuron H interacting with axon terminal of neuron F. So the space here, the synapse between F and H, the space between E and C, that's a synapse, and that synapse is axo-axonic. So axo-axonic synapse, that one, and this one. Yeah. So in both cases, axon C, the presynaptic neuron, axon F, the presynaptic neuron, is being affected. If the presynaptic neuron is induced to release more neurotransmitter, we call it presynaptic facilitation. If the presynaptic neuron is induced to release less neurotransmitter, we call it presynaptic inhibition. And this time is truly the last slide. Once a neurotransmitter has done its job, it is important that messenger be killed. It is important that the messenger should be terminated in order for the neurons to recognize a signal. All incoming signals should be new. So all old signal should be get rid of. So there are three mechanisms to get rid of the chemical messenger. The first one is called done by enzymes. So enzymatic degradation. For example, ACH, acetylcholine, ACH. There's an enzyme called acetylcholine esterase. Should be. And that enzyme cuts the acetylcholine into two pieces. Acetic acid, AA stands for acetic acid, plus choline. Okay, CHOLINE. Acetic acid here is the same as acetate. Acetate is the salt of the acetic acid, right? So you can say acetate or you can say acetic acid. And what is important is the choline is recycled. It's picked up by the axon terminal that releases it. So that choline is reused to make more acetyl-CoA, ACH. And so that's one example, enzymatic degradation. Another mechanism is the neurotransmitter is recycled by the presynaptic neuron that releases it. We call that presynaptic reuptake. Right? The neurotransmitter is picked up by the axon terminal of the presynaptic neuron. For example, serotonin. So the presynaptic neuron reuptake is happening in the case where the neurotransmitter involved is serotonin. And in fact, Prozac is a medication that prevents serotonin reuptake. So it prolonged the stay of serotonin in the synaptic cleft to increase synaptic transmission, to alleviate affective disorders such as depression. So Prozac, you now know, is a mechanism is basically preventing the presynaptic neuron from reuptaking serotonin, allowing serotonin to stay longer in the synapse, to activate the serotonin receptor, to basically maintain synaptic transmission, to alleviate depression. That's an application right there. And then the third mechanism, spillover. This mechanism is the simpler of the three. It means the neurotransmitter gets out, spills out of the synapse. If it spills out of the synapse, there's no receptor for it to affect anything. And so the neuroglia will pick the neurotransmitter to basically cleanse the site. So spill over, once if the neurotransmitter spills out of the synaptic cleft, if they spill out of the synapse, then the glial cells or the neuroglia, the support cells, will pick them up. And this is truly the end of today's recording. So on that note, I'm gonna end here, and then we'll talk again during the next recording. In the meantime, stay safe, be healthy, be happy, study well. Take care.